So Unit 6 talks about microbial genetics. It's going to be on your third exam, not your second one, but it's on the third one. So there's still some time to work with this unit. So when we talk about microbial genetics, obviously we're more focused on things like bacteria, but you'll see that a lot of the things that we talk about, you probably also heard in Gen Bio, okay, like transcription, translation, DNA replication, even the same enzymes to some extent, and so forth, all right? So hopefully, a lot of these things will be familiar to you. If not, don't worry, we'll go through it. The first thing is to look at DNA. The DNA nucleotide, take a look and see what it's made up of. The actual nucleotide itself is made up of deoxyribose, is the pentose sugar for DNA. Deoxyribose, that means it's actually a ribose without the oxygen. In which carbon?
The second carbon.
The second carbon. So if you look at the pentose sugar in DNA and the pentose sugar in RNA, you'll realize that the only difference is in the pentose sugar in RNA, which is the ribose, it has an oxygen in the second carbon. In the pentose sugar of DNA, it does not have that oxygen, and that's why it's called deoxyribose. It is a ribose without the oxygen. Okay, now if you look at the first carbon, it doesn't matter which one you're looking at. The first carbon of the DNA nucleotide or the RNA nucleotide, that's the attachment point for the nitrogen base. Now there are 4 different bases in DNA. Adenine, guanine, cytosine, and what's the 4th one? Thymine. Very good. So in DNA, the choices are adenine, guanine, cytosine, thymine, AGCT. In RNA, there are also 4 choices, but it's adenine, guanine, cytosine, and instead of T, it's uracil. So again, there are 4 choices that can be attached here, A, G, C, or U. But it doesn't matter if it's DNA or RNA, they're both attached at the 1' carbon. Okay, the 3' carbon in both of them have hydroxyl group. That the hydroxyl group in the 3' carbon, as well as the phosphate group in the 5' carbon, they're both very important in building the DNA, in attaching the nucleotides together to make the DNA, or the nucleotides together to make the RNA. So if you want to make DNA or you want to make RNA, these 2 groups, functional groups, the 3' hydroxyl and the 5' phosphate, are involved in making what's called the phosphodiester linkage, the bond that holds 2 nucleotide monomers together. So watch over here, please. This, for example, this part here of the marker could be the OH group. This OH group, for example, for the DNA. So here it is, OH. That's this part, OH. And this could be the phosphate group. The cap is the phosphate group. Now, what's going to happen is a water molecule will be released as a result of this reaction between the phosphate and the OH. So H2O, water is going to be released. We call that dehydration synthesis. So obviously an enzyme is involved in this too. So an enzyme is going to grab onto this nucleotide and grab onto this nucleotide and force the attachment. Watch. Force the attachment. So the OH of this nucleotide is going to react with the phosphate of the incoming nucleotide, and it's going to do this. That's a phosphodiester bond. The result of this is water is going to come out of this. H2O, water, and that's why it's called dehydration synthesis. So what did I just create? I created a dinucleotide. One nucleotide, two nucleotides, dinucleotide with a phosphodiester bond. That's a covalent bond. And I can do it again and again and again and again and again to make the DNA. So that's a quick review of your Gen Bio there. Hopefully you remember that. Now, when it comes to the bases, the adenine and the guanine are referred to as purines. And you have to remember that. So one of my students a while ago, she said, I know how I remember it, and you can share it with your students. She says, pure as gold. Nice. Purine as adenine, gold, guanine. So the A and the G are purines. And a purine will have two cyclic structures like this. You don't need to memorize the structure. I'm just showing you that's why they're called purines. You have two cyclic structures. Now, a pyrimidine has one cyclic structure, and it's not the adenine or the guanine, because the adenine and the guanine are the purines. So the pyrimidines are the CUT. I actually, when I was a student, I remembered it as cut pyrimidine. CUT are the pyrimidines. But if you want to know it as pure as gold, you can do that too. Or you can know both of them. Cut pyrimidine and then pure as gold. So obviously, when it comes to the pyrimidines, the C and the T are in the DNA, and the C and the U are in the RNA. Everybody okay so far? Good. Here we are. This is what I was trying to show you with the markers in my hand. How the DNA is built into how the nucleotides come together to build the DNA. Now, terminology. The sugar with the base, just this part, is called the deoxyadenosine, because that's an adenine, and that's the deoxyribose. So it's a deoxyadenosine, just the sugar and the base. When you add one phosphate to it, then it becomes deoxyadenosine monophosphate. When you have two phosphates, it's deoxyadenosine diphosphate until you get to deoxyadenosine triphosphate. That's the meaning of those things when you run into them in your readings and stuff. Now, if you have a cytosine up there, then it's deoxycytosine monophosphate. In this case, it's a deoxyribose with a cytosine and a phosphate. That's what that means. And now you're attaching them together. So here's your dehydration synthesis. Now this needs energy. This reaction needs energy. The energy is already built into this area here. The energy that you see here, that's the energy that is supplied into the system. So this molecule comes with the monomer, right? And it also comes with a bag of energy because it knows this is anabolic. Means I need energy. And it comes with the energy. And that's what this guy is telling you. PPI is this part right here. When these two come off, you have to break this bond. To release those two phosphates, you have to break this bond. And when you break this bond, you release the energy that drives the reaction. And now you have it attached to each other.
So what's the name of this energy?
It's just energy. It's Gibbs free energy.
Uh-oh.
So that's what keeps you alive. All that energy that's harnessed in those high energy bonds is what's keeping you alive. Now, if we look at DNA, we find that DNA is double-stranded. Now, remember in viruses, that's true for the most part. But when we come to viruses, they tend to break the rule. Yeah, but it's true for the most part. DNA is double-stranded. RNA is single-stranded, and it's true for the most part, unless you are talking about certain viruses. And here's the base pairing. G with C, A with U. So every G faces a C. And it doesn't matter if it's RNA or if it's DNA. But when you're talking about adenine in RNA, it faces uracil. In DNA, it faces thymine. Oh, wait a minute. On one strand of the DNA, yeah? Watch this. On one strand of the DNA, there's a purine. Adenine is a purine, pure as gold. Adenine is a purine. And it will always face a pyrimidine. Thymine is a pyrimidine. And it's always that way. You will never have a purine facing a purine. And if you do, that's a genetic mutation that needs to be fixed. So watch this. A purine facing a pyrimidine. A purine facing a pyrimidine. In this case, a purine facing a pyrimidine. All the time. Even in RNA. A purine facing a pyrimidine. A purine facing a pyrimidine. Uracil is a pyrimidine. And that kind of question tends to sneak in an exam easily. But if you're not paying attention to the pictures, you're not going to know what we're talking about. But it's always the case. On one strand of the DNA, it's going to be a purine, either A or G. That means on the other strand of the DNA, just across the hall there, it has to be a pyrimidine. Now the question now is, what bond is holding the two strands together in DNA? And what is this dotted line?
Hydrogen.
That's a hydrogen bond. Yeah. It's going to be a hydrogen bond. So you got to understand the difference between these two. The bond that's holding the strands together, those are covalent bonds. Those are phosphodiester bonds. But when I'm talking about the hydrogen bond, I'm talking about the bond that's holding the two strands together. That's different. That's a hydrogen bond. One of them is sensitive to heat. The other one isn't. Which one is it? Which one is sensitive to heat?
The phosphate.
No. The phosphodiester bond needs an enzyme to break it.
Heat, hydrogen bond?
It's the hydrogen bond.
That's it.
So if I add heat to your DNA, watch this, here's your DNA. I add heat. I'm going to do this. I'm going to separate the two strands. I'm not going to break the strand. I'm going to separate the two strands. But what if I want to break the strand? I need an enzyme and water.
Yeah.
Because what's the name of that reaction?
Hydrolysis.
Hydrolysis. So there's a difference between breaking the strand into nucleotides and separating the two strands. When you separate the two strands, you still have two strands.
Which would be the hydrogen.
That's the hydrogen bond, right? That's the one that needs heat. You add heat, you separate the two strands, but you still have the two strands. But if you want to break the strand into its monomers, the nucleotides, you need an enzyme and water. Now you're breaking it. Same thing for RNA. The bonds that's holding the RNA structure. Now RNA is single-stranded. It's just folded. It's also hydrogen bonds. So again, heat will break those down too. But what it will do is it will unfold this thing. It's like you taking a bow and unwrapping it, unfolding it. You still have the part of the bow, the ribbon. But if you want to cut the ribbon, you got to take scissors and cut it. You see? That's an enzyme. So that would also be hydrolysis too. Now we're gonna talk about something that's very important that every bacterial cell and every organism does right before they decide to multiply. They copy the DNA. In eukaryotic cells, we call that the S phase of the cell cycle, which stands for synthesis,
That's right
and
before
right before division of the cell. So the first thing your cell is going to do when it divides is to multiply the DNA, is to duplicate the DNA, copy the DNA. Bacteria is the same. I mean, right before it undergoes binary fission, it's going to copy its DNA too. Why? Because at the end, I'm going to have two cells. And they both need a copy of the DNA. But at the beginning of the process, I only have one copy. And if I don't copy the DNA, then what's going to happen is one cell will have a copy, the other one won't. What's going to happen to the one that doesn't have a copy? It's dead. It won't know what to do. Doesn't have DNA. It won't know what to do. So the first thing that happens is copy the DNA. Then divide the cell. So we're going to talk about DNA replication. This alone is a separate chapter in Gen Bio. The copying of the DNA has to start somewhere. And that somewhere is called the origin of replication. So there's a sequence on the DNA, a specific sequence on the DNA that enzymes, as they're floating by, they say, oh, there's the origin of replication. So it's just sequence based. A lot of these things are sequence based. So it's got that sequence that the enzyme binds to. Which enzyme? Topoisomerase. So topoisomerase 1, as it's floating by, it will recognize, oh, that's the origin of replication, and that's where I need to sit. And it will. It will sit on the origin of replication, and look what it does. It bubbles out the DNA by breaking down the hydrogen bonds right here. See, these are hydrogen bonds. Break them down. And then it leaves. I'm done. And it leaves. And what does it leave behind? It leaves this bubble here that's referred to as the replication fork. And there are two of them. When it bubbles, look up here, please. This V-looking shape here, going this way, is a replication fork. You can see it better here. That's a replication fork. And this one here is another replication fork going that way. So it creates two replication forks going in both directions. Now there's a reason behind it. Because DNA replication is bidirectional. You really think this DNA is going to be copied in one direction while the other side waits? I wonder when he's going to finish. No. It's going to do the copying in both directions at the same time. Your cell is very efficient. So a whole bunch of enzymes are going to sit here and they're going to go this way. At the same time, a whole bunch of enzymes, the same ones, are going to sit here and they're going to go this way. Now I want you to do something. I want you to pretend, look up here please, that there is a mirror right in the middle cutting through here, right here. Cutting the origin of replication in half. You see that? But this is a very weird mirror. Why? I know I have a mirror image because it's a mirror, but whatever is happening here is going to happen here. It's one of those weird mirrors that flips you upside down. So whatever is happening here is going to happen here, and whatever is happening here is going to happen here. And what's happening here? Listen carefully. I am making the leading strand. Why is it called the leading strand? Because I'm going to make this piece of DNA, yeah, in one continuous motion. I'm just going to keep going until I fall off the end. At the end of this process, you're going to have one piece of DNA all the way to the end, and the same thing is going to happen with this one. One piece of DNA. Because you're making it in one piece, that's called continuous. Compared to this one. This one, look at it. It's made in pieces. So, this one will say, well, I can't make the third piece until you open it up. So it's going to have to wait until you open this up, and then it makes the third piece. Because it's going to make the pieces going this way. But you haven't opened it up yet. So it's going to take a little bit longer. That's why this is called the lagging strand, the one that's slower. The leading strand, it's really fast. The leading strand has to actually wait for the lagging strand to finish its job. If it was up to the leading strand, it would just go right there. But it's going through, and I'm like, okay, we have to wait. Okay, you're done. Okay, now, okay, are you done now? Okay, and it keeps going. So the leading strand is made in one continuous motion. The lagging strand is made in pieces. Does anyone know the name of the pieces?
Okazaki.
Okazaki fragments, right here. You gotta pay attention now. Watch this. The leading strand is made going towards the fork. Trying to help you out. The leading strand is made going towards the fork, whether it's this one or this one. Now, you can say the lagging strand is being made going towards the origin of replication. The lagging strand is being made going towards the origin, or you can say away from the fork. It's up to you how you want to remember it. Now watch this. Because, say?
I'm sorry, being made going towards the origin or?
Yeah, you can say the lagging strand is being made going towards the origin, or you can say, depends on your point of view, away from the replication fork. Now, because the lagging strand is made in pieces, that's called discontinuous. The leading strand is continuous, means in one piece. The lagging strand is discontinuous because it's made in pieces, and the name of the pieces? Okazaki fragments. All right, now at the end of this process, you'll notice something. We're going to get into this in a little bit more detail. But at the end of the process, you're going to notice something. They will both look the same. You won't know which one came from what. Because the pieces, the Okazaki fragments, will be ligated together. And they will both, at the end, look the same. After the whole process is done. Now, I want you to pay attention to something. At the end of this DNA replication, the original template is blue. This is our template. This is our original DNA. But look at the copies. They are half original, right? And the orange part you just made. Half new. Half old, half new. What is the name of that? Half old, half new. It's on the screen.
Semi-conservative?
Semi-conservative. DNA replication is semi-conservative. The product of DNA replication, the copy, is always going to be half old, half new. Why? Because it's so much easier to do. Watch this. Here's your double-stranded DNA. Watch now. 5' phosphate, 3' hydroxyl. Sounds good? The other strand is going to go the other way because DNA is anti-parallel. They go in opposite direction. 5' phosphate, 3' hydroxyl. Sounds good? Yeah. Watch me copy it. Watch. Open it up. Copy. I'm done. So when I open it up, when I copy this one, I'm gonna have half old, half new. Am I right? And when I copy this one, I'm gonna have half old, half new. What is that called? Semi-conservative replication. So you got a whole bunch of words thrown at you. Anti-parallel tells you the direction of the 5', 3' phosphate of DNA, that they're not parallel, they're not like this. They are parallel, but they go in opposite direction with respect to the 5' and the 3'. By the way, what's on the 5'? What's on the 5'?
Phosphate,
A phosphate. What's on the 3'?
hydroxyl group.
Hydroxyl. Very good. Now, the other thing I threw at you is leading strand versus lagging strand. Leading strand is made in one motion. What is that called? Continuous. The lagging strand is made in pieces. What is the name of the pieces?
Discontinuous.
The name of the pieces. Okazaki. The name of the process. If this was continuous,
Discontinuous.
this is discontinuous. Good. Which one of the two, continuous or discontinuous, or leading or lagging, is going towards the fork?
Leading.
The leading strand. Very good. At the end of the whole thing, you're going to have a DNA copy, and the molecule will be half old, half new. What is the name of that again? Semi-conservative. There you go. Ooh. Let's take a look at this. This is showing you what's happening with all these enzymes. It looks overwhelming, and it's not that bad. What's happening with the enzymes right after the first topoisomerase comes in and opens up the DNA to create the replication forks. Am I right?
Mm-hmm.
So here's a replication fork right here. And here's another one. And here's that imaginary mirror that I told you about. And I said anything that's happening here is going to be happening here. And anything that's happening here is going to be happening here. All right, here we go. Make a list of them. The first enzyme is topoisomerase. Don't worry about the number. Topoisomerase. That's the one that's going to sit at the origin of replication to create the replication forks. So let me do it again. Number one, topoisomerase. Topoisomerase. Don't worry about which number it is. Just the topoisomerase. It will sit at the origin of replication. It's usually abbreviated ORI. Origin of replication. And what will it do? It will create the two replication forks. It will create this bubble, resulting in the production of the two replication forks, one going this way and one going that way, because DNA replication is bidirectional. Okay. Then what happens? Then what happens is I remember my mom. That's what happened. How do you remember your mom in the middle of this? Because my mom used to tell me, and not that I listened, untie your shoelace. It makes taking your shoes off a lot easier. Did I listen? No. But you better listen to this one. Untangle the DNA before you start copying it. See, the DNA up here is all tangled up. Same thing with here. This one you cannot ignore. You have to untangle the DNA before the next enzyme goes through. Otherwise, it's gonna hit a wall. I can't deal with this. It's another topoisomerase that does that. So number 2 on your list is a different topoisomerase. Now listen to the words now. The second topoisomerase there will come in, listen, sit ahead of the fork. Because that's where it is. It's ahead of the fork. On this side and this side. And again, whatever is happening here is happening here. So it's sitting ahead of the fork. To do what? To untangle the DNA. So it's doing this to the DNA. Untangle, untangle, untangle as it's moving through. All right, the next enzyme that comes in is this guy right here, helicase. Helicase is gonna sit at the fork. And that's what it's showing you here. It will sit at the fork. And now, watch what it's gonna do. The double-stranded DNA, it's gonna break the hydrogen bonds. You really think we're gonna use heat in your cells? I mean, I can, but you won't survive that. It's enzymes. So in the case inside your cells, we're not gonna use heat to break hydrogen bonds. We're gonna use an enzyme, in this case, helicase. So helicase is gonna come in, sit at the fork. Hopefully you're writing these things down. And unzip the DNA. Unzip the DNA by breaking down the hydrogen bonds. That's exactly what the picture is showing you here. Helicase is gonna sit at the fork and it's basically taking your double-stranded DNA, right? And making it single-stranded by breaking down which bonds? The hydrogen bond. So we're not going to use heat here because you would need a lot of heat to do that and you won't survive that. All right, now I made the DNA single-stranded. I haven't copied anything yet. Something has to come in to prevent the DNA from rezipping. Because if helicase drives by and the DNA goes, is he gone? Yeah, he's gone. Let's rezip. You're not going to copy anything. So something has to come in to keep the DNA single-stranded after helicase has already left. And it's this guy right here. It's called single-stranded binding protein, SSBP. The single-stranded binding proteins, listen to the word, they recognize single-stranded DNA. They don't bind to double-stranded DNA. I don't know what that is. I only bind to single-stranded DNA. So that's why they come in after the helicase has had a chance to single-strand your DNA. Then they come in. These are not enzymes, they're just proteins. They come in and bind to the DNA. That's all they do. And what they're trying to do is they prevent them from re-zipping, which means they prevent them from reforming hydrogen bonds. And then the fun begins. The first site of copying happens right here when the next enzyme comes in, which is primase. The next enzyme that comes in is called primase. Here it is, the word primase. Now look over here. Whatever is happening here is also happening here. And whatever is happening here is also happening here. So now you have 4 primases coming in. One sitting here, and it will go this way. And one sitting here, and it will go this way. And one sitting here will go this way, one sitting here will go this way. But these guys are weird. Even though we're trying to make DNA, we're trying to copy DNA into DNA. It starts out as RNA. You gotta be careful with that. The primase makes an RNA primer. This thing that it's making, that's RNA, not DNA. Small piece of RNA. I don't know, let's say 20 nucleotides. Small piece of RNA. Small piece of RNA, small piece of RNA. So let me try this. If the primase reads a G on the DNA, what will it bring on the RNA? C. Good. If the primase reads a C on the DNA, what will it bring on the RNA? G. If the primase reads an A on the DNA, what will it bring on the RNA? U. Very good. Nice catch. That's what I want to get to. You're not going to have T's here because it's RNA. So you're going to have yourself. Now, why are you doing this? Why do you make the RNA? Because of the next enzyme. The next enzyme that's going to come in, it's called DNA polymerase 3, is very weird. It's weird how it needs to know where it needs to start working. I mean, it's very good at what it does. DNA polymerase 3 is the enzyme that's going to make DNA. It's built into the name, DNA polymerase. I make DNA. And I'm very good at it. But you just need to tell me where I need to start to work. That's what this primer is doing. The primer, after it's laid down, this primase, you're going to see the next slide, is going to fly away. It's done. I'm leaving. The primer now will tell DNA polymerase 3 where to start. Start here. Watch this. Start here, where the 3' end is. So DNA polymerase 3, you'll see this, will come in and bind to the 3' end right here and start making DNA this way. Now, why? Why do you need that 3' end? Because what's at the 3' end? What's at the 3' end? Hydroxyl. Good. Watch this over here. Look at this over here. Here's the primer. Let's say this is the RNA primer. Okay. What's at the 3' end? Hydroxyl. Ooh, hang on. I pushed it. Let me try it again. Am I back to the... Here it is. What's at the 3' end again? Hydroxyl, right? Watch this. There. DNA polymerase 3 is going to come in and bind to this 3' end right here, so that it will take the DNA nucleotide and attach it there. You see that? Now let's see. That means this primer told DNA polymerase 3 where to start, yeah? And it also gave it the hydroxyl group to be able to do what? To be able to attach the first nucleotide. Do you agree? Good. If you don't agree, let me show you what would happen without the primer. Watch what would happen without the primer. There's no primer there. Imagine, pretend as if there's no primer there. DNA polymerase 3 is going to come in with the DNA nucleotide, and it's going to go, I don't know. Where does he want me to start? I don't know. I don't know what I need to start, but let's say here. Okay, here. Just by chance. I will come in, bind my first nucleotide. Watch. And it will fall apart. Because there's nothing to attach it to. So, that primer does 2 jobs. Nice question on the exam. That primer does 2 jobs. Number 1, it tells DNA polymerase 3 where to start. And it also provides it the hydroxyl group that it desperately needs to attach the first nucleotide to. So let's try this. Watch. Let's make the blue the primer. Here it is. The blue is the RNA primer. Good. Anything other than that is just DNA. Here we go. Watch this. DNA polymerase 3 is going to come in, bind the first nucleotide. Then it's going to come in and bind the second nucleotide. And then it's going to come in and bind the third nucleotide. Am I right? Do I need the RNA primer anymore? Do I still need this? No. Another primer is going to come in and do this. Watch, watch. I don't need this anymore. That's DNA polymerase 3's cousin. DNA polymerase 1 will remove the primer and replace it with DNA. So at the end, you're only going to have DNA. So let's see it. So here are your primers. Watch this. The topoisomerase is still doing this, untangling the DNA. This is happening all at one time. The helicase is still unzipping the DNA. The single-stranded binding proteins, some of them are coming off and some of them are coming on. Right? Here's another primase coming in to build the new primer, while this guy, primase, is already leaving because it already finished the primer. Same thing with this one. This one is already leaving because it finished the primer, while a new primer is being made here and here. This guy already finished the primer, so it's leaving. But look who's arriving, DNA polymerase 3. Look where it's binding at the 3' end. At the 3' end, at the 3' end, 3' end. What is it about to make? DNA polymerase 3 makes, what is it? DNA polymerase 3 makes DNA. There you go.
Starts copying the DNA.
Yeah, it's just start copying the DNA. Watch it happen. Watch this now. There it is. All right, this piece here already built. That's the yellow is the DNA. Here's another piece. DNA polymerase 3 is doing a very good job, and as it's moving through, it's knocking off the SSBPs. It's also following helicase as it's unzipping, and helicase is following topoisomerase as it's untangling. This is amazing stuff. This happens really fast too. Meanwhile, this guy here is being made in pieces. What's the name of the pieces? Okazaki fragments. Here's one piece of Okazaki fragment, and we're about to build the next piece right here. Let's see. There it is. This is done. This Okazaki fragment is done. And this piece is still going. This piece is done. And what's going to happen is something is going to come in to remove the primer. Here it is, DNA polymerase 1. I don't need that primer anymore. So DNA polymerase 1 is going to come in and replace the primer with DNA. So you're not going to see the red anymore. It's all going to be yellow. Meanwhile, this guy is about to start making the next Okazaki fragment. And so is this one. You know how long it took me to draw these things? Very long time. All right, here we go. This piece is complete Okazaki fragment. That's completely DNA. And this one is almost done. The DNA polymerase 1 is binding to remove the primer. And then you realize something. There's a gap between this piece and this piece that you have to seal. Otherwise, you're going to have broken pieces of DNA. And that's the function of the last enzyme, DNA ligase. There it is, a complete piece right there. After DNA ligase does its job. Are we good? Yeah, it takes some study, I know. The final thing I'm going to say is this. In bacteria, or prokaryotes, the DNA is circular. Remember us talking about that. So you have the bacteria has one origin of replication. And the DNA gets copied like this. Watch. In a circle. Like that. until you get to the other side, because it's bidirectional and it's a circle and can do this. In our chromosomes, you can't do that. Number one, they're linear. Number two, they're very long. See, this one, because they're circular and relatively smaller than ours, they can do this. They can afford to do this. Our DNA, however, is quite long and linear. So if you rely on one origin of replication, you're gonna wait quite a while to copy your DNA. So that doesn't happen. In eukaryotic DNA replication, you have multiple origin of replications, creating multiple replication forks and replication fork bubbles. And then you're gonna make the pieces that you're gonna eventually rely on ligating them together to make the full piece. That's one main difference between eukaryotic and prokaryotic DNA replication. One origin of replication, multiple origin of replications. The main reason why this one has one, because it's circular and smaller. The main reason you need multiple ones, because they're linear and much longer. Sounds good? All right, I'll leave you with this until I see you on Tuesday.